In September 1928, Alexander Fleming returned to his laboratory at St. Mary's Hospital in London after a 2-week vacation in Scotland. His laboratory was notoriously cluttered, culture plates stacked haphazardly on benches and windowsills, some days or weeks old. Fleming was preparing to clean up, discarding contaminated plates, when he noticed something unusual on one Staphylococcus culture plate. A blob of mold had contaminated the dish, and around the mold the bacterial colonies had dissolved. Where the mold grew, the bacteria died. Fleming examined the plate closely, recognizing that the mold was producing something that killed bacteria. He isolated the mold, identified it as Penicillium notatum, and named the antibacterial substance it produced penicillin. On February 13, 1929, Fleming presented his findings to the Medical Research Club. The audience listened politely and asked no questions. No one seemed particularly interested. Fleming published his discovery in the British Journal of Experimental Pathology in May 1929, describing penicillin's antibacterial properties and suggesting potential therapeutic applications. The paper attracted almost no attention. Most researchers who read it assumed penicillin was simply another antibacterial substance, like lysozyme, which Fleming had discovered years earlier. Lysozyme killed bacteria in the laboratory but was ineffective in the body, and scientists assumed penicillin would prove similarly impractical. Fleming tried to purify penicillin with help from chemists at the London School of Hygiene and Tropical Medicine, but they failed. The mold produced penicillin in tiny quantities. The substance was unstable, and extraction techniques were crude. After months of frustration, Fleming's collaborators declared that large-scale production was impossible. Fleming, disappointed but not surprised, essentially abandoned penicillin research by 1932. Alexander Fleming was born on August 6, 1881, on a farm in Lochfield, Scotland, the third of 4 children from his father's second marriage. His father died when Alexander was 7, and the family struggled financially. At age 13, Fleming moved to London to live with an older brother and attend school. He took a job as a shipping clerk, but disliked the work. In 1901, when he inherited a small sum of money, his brother suggested he study medicine. Fleming enrolled at St. Mary's Hospital Medical School, excelling in his studies and graduating in 1906. He joined the research department rather than practice clinical medicine, attracted by the intellectual challenge of understanding disease mechanisms. Fleming's early research focused on immunology and chemotherapy, the use of chemicals to kill pathogens without harming patients. During World War I, he served in the Royal Army Medical Corps, working in field hospitals, treating soldiers with infected wounds. He observed that antiseptics, widely used to disinfect wounds, often did more harm than good. Strong antiseptics killed surface bacteria but couldn't penetrate deep infections, and they destroyed white blood cells that fought infection naturally. Fleming concluded that better treatments were needed, substances that could kill bacteria without damaging human tissue. After the war, he returned to St. Mary's Hospital and resumed research, searching for natural antimicrobial agents. In 1922, Fleming discovered lysozyme, an enzyme found in tears, saliva, and mucus that dissolves certain bacterial cell walls. It was an important finding, demonstrating that the human body produces natural antibacterial substances, but lysozyme was effective only against harmless bacteria, not the pathogens causing infections. Fleming's discovery of penicillin in 1928 seemed more promising. Penicillin killed Staphylococcus, Streptococcus, and other dangerous bacteria without harming human cells. But Fleming couldn't purify penicillin sufficiently to test it in patients, and without clinical trials, the medical community dismissed it as a laboratory curiosity. Fleming continued using crude penicillin in his lab to inhibit bacterial growth in cultures, but he abandoned attempts to develop it as a medicine. For the next decade, penicillin languished forgotten. Fleming occasionally mentioned it in lectures, but few paid attention. In 1936, at the Second International Congress of Microbiology in London, Fleming presented penicillin research to an audience of prominent bacteriologists. No one expressed interest, no one asked questions, no one saw therapeutic potential. Fleming's colleague V.D. Allison later remarked that Fleming was keenly disappointed but remained convinced penicillin was important. By the late 1930s, however, even Fleming had mostly moved on to other research. Penicillin remained a footnote in scientific literature, a curious observation with no practical application. The discovery that would save millions of lives was ignored. In 1938, Howard Florey became professor of pathology at Oxford University. Florey, an Australian-born physician and physiologist, was interested in naturally occurring antibacterial substances. He assembled a team to study lysozyme, hoping to understand how enzymes killed bacteria. In 1939, Florey hired Ernst Chain, a German Jewish biochemist who had fled Nazi Germany, to conduct a literature review of antibacterial substances. Chain read Fleming's 1929 paper on penicillin and found it intriguing. Unlike lysozyme, penicillin killed pathogenic bacteria. Chain proposed that Florey's team investigate whether penicillin could be developed as a medicine. Florey agreed, and in 1939 the Oxford team began growing Penicillium mold and extracting penicillin. The timing was significant. World War II began in September 1939, and Florey recognized that bacterial infections killed more wounded soldiers than injuries themselves. If penicillin could treat infections, it might save thousands of lives. But producing penicillin was extraordinarily difficult. The mold grew slowly, yielding tiny amounts of the antibiotic. Chain and biochemist Norman Heatley developed techniques to extract penicillin from the culture broth, but the process was inefficient. The Oxford team needed hundreds of liters of mold broth to produce enough penicillin for experiments. They grew mold in every available container: bottles, flasks, bedpans, baths, and ceramic vessels. The laboratory became a penicillin factory, staffed by penicillin girls who tended cultures and collected broth. By early 1940, the Oxford team had accumulated enough penicillin to test its effectiveness in mice. On May 25, 1940, Florey injected 8 mice with lethal doses of Stretptococcus bacteria. 4 mice received penicillin injections. 4 received no treatment. Within 16 hours, the untreated mice were dead. The penicillin-treated mice survived. Florey repeated the experiment with similar results. Penicillin worked. It protected animals from bacterial infections without causing harm. The team rushed to produce enough penicillin for human trials, but production remained achingly slow. Each patient required doses extracted from hundreds of liters of mold culture. In February 1941, Albert Alexander, a 43-year-old policeman, became the first person treated with penicillin. Alexander had scratched the corner of his mouth while pruning roses. The wound became infected, and despite sulfa drugs, the infection spread. Abscesses developed on his face, scalp, and lungs. His eye was removed. Doctors expected him to die. On February 12th, Florey injected Alexander with 200 milligrams of penicillin, followed by additional doses every 3 hours. Within 24 hours, Alexander's fever dropped. Abscesses began healing. After 5 days, his condition improved dramatically. But on the 5th day, the Oxford team ran out of penicillin. They had collected and recycled penicillin from Alexander's urine, extracting every molecule they could recover, but it wasn't enough. Alexander relapsed and died on March 15th. Despite Alexander's death, the trial proved penicillin worked in humans. Subsequent patients treated with adequate supplies recovered completely from infections that were otherwise fatal. The challenge was producing penicillin in sufficient quantities. The Oxford team approached British pharmaceutical companies, but wartime conditions made large-scale production difficult. Factories were bombed. Resources were scarce. In 1941, Florey and Heatley traveled to the United States to seek American support. They met with officials at the U.S. Department of Agriculture, pharmaceutical companies, and research institutions, demonstrating penicillin's effectiveness and requesting assistance with mass production. American companies, particularly Pfizer, developed deep tank fermentation methods that dramatically increased yields. By 1943, penicillin was being produced industrially. By 1944, enough was available to treat Allied soldiers on D-Day. Penicillin transformed wartime medicine. Infections that had killed soldiers for centuries became treatable. Pneumonia, gangrene, septicemia, and wound infections that once meant death or amputation could be cured with penicillin injections. Doctors observed recoveries that seemed miraculous. Soldiers near death from infection walked out of hospitals weeks later, fully healed. The drug was so effective that commanders worried about soldiers using penicillin to treat venereal diseases, potentially encouraging risky behavior. Penicillin saved thousands of lives during World War II, and its success sparked a revolution in medicine. Researchers began searching for other antibiotics, eventually discovering streptomycin, tetracycline, and numerous others. The antibiotic era had begun. In 1945, Fleming, Florey and Chain shared the Nobel Prize in Physiology or Medicine for the discovery and development of penicillin. The award recognized Fleming's initial observation, Florey's leadership in clinical development, and Chain's biochemical work purifying the drug. Fleming became famous, celebrated in newspapers and magazines as the man who discovered the miracle drug. Florey and Chain, who had done the difficult work of turning Fleming's observation into a practical medicine, received less public recognition. Fleming, modest by nature, repeatedly credited Florey and Chain, but the public preferred the narrative of the lone scientist making an accidental discovery. Fleming spent his later years lecturing on penicillin, warning that overuse and misuse would lead to antibiotic resistance. In a 1945 interview, he predicted that bacteria would evolve resistance if penicillin was used carelessly. His warnings were prescient but largely ignored. By the 1950s, penicillin-resistant Staphylococcus strains were emerging in hospitals. Today, antibiotic resistance is a global crisis, threatening to return medicine to the pre-antibiotic era. Fleming understood that penicillin was not a permanent solution, but a tool that required careful stewardship. His discovery saved millions, but it also created new challenges that persist decades later. Fleming died on March 11, 1955, at age 73 from a heart attack. He was buried as a national hero in St. Paul's Cathedral, an honor reserved for Britain's most distinguished citizens. His discovery of penicillin is celebrated as one of the greatest medical breakthroughs in history, saving an estimated 200 million lives since its introduction. Yet Fleming's contribution was serendipitous observation, not systematic development. He noticed mold-killing bacteria, recognized its significance, published his findings, and tried unsuccessfully to purify it. When he failed, he moved on. It was Florey and Chain who transformed Fleming's curiosity into a medicine, yet they remained less famous. The story of penicillin illustrates how scientific progress depends on both discovery and development. Fleming made the crucial observation, but without Florey and Chain, penicillin would have remained a forgotten laboratory phenomenon. Florey and Chain provided the systematic research, chemical purification, animal testing, clinical trials, and industrial production that made penicillin a practical medicine. Both contributions were essential. Neither alone would have produced the miracle drug. Yet history celebrates Fleming's accidental discovery more than Florey and Chain's deliberate development, reflecting a cultural preference for eureka moments over patient, methodical work. Penicillin's impact extends beyond medicine. The antibiotic revolution that penicillin launched increased human life expectancy dramatically. Diseases that killed routinely, pneumonia, tuberculosis, syphilis, became treatable. Surgeries that had been too risky because of infection became routine. Childbirth became safer. Penicillin and its successors transformed public health, enabling population growth, longer lives, and reduced infant mortality. The drug that Fleming discovered by accident became foundational to modern civilization, as essential as clean water, vaccines, or electricity. The 12-year gap between Fleming's discovery and penicillin's widespread use demonstrates how scientific breakthroughs often require years to translate into practical applications. Fleming published in 1929. Florey and Chain began work in 1939. Mass production started in 1943. Penicillin saved lives on D-Day in 1944, 16 years after Fleming's original observation. During those years, millions died from infections that penicillin could have cured. The delay resulted from technical challenges, lack of funding, institutional inertia, and the absence of a compelling reason to invest in development until wartime necessity created urgency. War accelerated progress, but peacetime indifference had allowed a life-saving discovery to languish for over a decade. Fleming's penicillin story parallels others throughout history. Watson Watt's radar was dismissed until it saved Britain. Mitchell's Spitfire was mocked until it defended the skies. Brunel's ships were called extravagant until they connected continents. Harrison's chronometer was rejected until it solved navigation. Semmelweis's hand-washing was ridiculed until it saved mothers. The Wright brothers were ignored until their planes changed warfare. Goddard's rockets were mocked until they reached space. Hillman's vaccines broke rules until they saved millions. Borlaug's wheat was opposed until it prevented famine. Marshall drank bacteria to prove ulcers were infectious. And Fleming's penicillin was forgotten until war demanded miracles. Each innovator faced indifference, skepticism, or hostility. Each was vindicated, often years later, when circumstances forced recognition. Fleming published his discovery, but no one cared. For 12 years, penicillin sat in scientific literature, mentioned occasionally, ignored consistently. Then war created urgency. Florey and Chain returned to Fleming's forgotten paper, and penicillin became the miracle drug that saved the world. The discovery was accidental. The development was deliberate. Together, they transformed medicine. Fleming's mold, contaminating a culture plate in a cluttered London laboratory, became the foundation of the antibiotic age. The scientist who returned from vacation to find his experiment ruined had actually discovered a way to save millions. But it took a decade, a world war, and 2 other scientists to prove it.